Hi everyone. In this video, we discuss about cell division cycle phases. So to understand the different stages of cell cycle phases, we should know how actually these different phases of cell cycle or cell division were discovered by scientists and what experiments that led to identify that there are different phases exist in cell division or cell cycle. Okay, initially mitosis or what we call it, the different phases of mitosis uh, were were observed by the scientists using the light microscope. So when we take the cell cycle, there we find another stage or another phase in cell cycle, that is called interphase. So there is a mitosis and a interphase. So this interphase, so how the different phases of interphase were observed by the scientists. So mitosis, by using light microscope, they observed different phases in mitosis, how prophase takes place, and how metaphase, anaphase, and telophase takes place by seeing the changes, the structural changes that takes place in the cell. When it comes to interface, the experiment that led to identify that different phases are present in cell division, that in interface is quite interesting. So they used one experiment. They conducted an experiment that is called a tritiated thymidine addition experiment. Okay, so different phases interface were observed by using a tritiated thymidine experiment. So to identify different phases in mitotic phase, they use light microscope. So how they observed that different phases are present in this interface of cells is so interesting, and also it needs some experimental analysis. Okay, here they took the HeLa cell lines. Okay, so they exposed all cells to the tritiated thymidine. Okay, so in the interface, actually, DNA synthesis takes place. We know all that. In the DNA synthesis takes place in the interface, and during this mitotic phase, we see the chromosome condensation and separation, segregation, and finally cytokinesis and all those things. Okay, so when they observed under light microscope, they did not observe the DNA synthesis in the mitosis. So they thought that there is this DNA synthesis takes place in the cells before they enter into this particular time. Particular phase, that is called the interphase. Okay, they called it interphase. That means interphase is a phase which is present between the two successive cell divisions, or no, we can say it's mitotic phases. Okay, so they exposed the cells to the this tritiated thymidine. When they exposed to tritiated thymidine, they thought that since the DNA synthesis takes place in the interphase, the cells which are in the interphase condition, that means they took the asynchronous cell culture. Some are also even the mitotic cells in that particular the cell plate. So they took the asynchronous culture. Asynchronous culture means some cells they just started their interface. Some they begin while some they're about to finish, and some even they might be in the even mitotic phase also. That means at different stages we find the cells in this cell culture. So they exposed these cells to the this tritiated thymidine, and they thought so cells which are all in this interface, they receive the tritiated thymidine because the thymidine gets incorporated during the DNA synthesis, and they thought that all the cells which are in the interface, since they synthesize DNA, they get this tritiated thymidine in their chromosomes. So when they observed under microscope, this interface cells, when they observed under microscope, they found that only few cells have this tritiated thymidine. For their surprise, very few cells have they tritiated them, not all the interface cells. Okay, when they exposed cells to thymidine and only few cells received the thymidine, they understood that okay, cells which are actually synthesizing their DNA in this 30 minutes of time, they incorporated this tritiated thymidine in their chromosomes. And also they understood that okay, that the remaining cells are not synthesizing the DNA but still in the interphase. So that means they are in the interphase only, but at a different about at a different state in the interface, but not they are synthesizing the DNA. That means they understood, okay, only at a particular time in that interface, the DNA is synthesized, not like a throughout the interface the DNA is synthesized by these cells. Okay. mm, so the time in which the DNA, the cell synthesizes their DNA in the interface, they named as a S phase. They named as S phase. Okay. So the rest of the cells, why they did not receive the tritiated thymidine? That means only few cells are at this state of synthesizing DNA, and the remaining cells which are not in the state of synthesizing DNA, maybe these cells are yet to start their DNA synthesis, or these cells might have completed their synthesis of DNA. That's why they did not receive the tritiated thymidine. So that what they understood. Okay, later they identified some other phases in the this interface, like like G2 phase and G1 phase, also understood by the scientists. That we'll discuss how they understood, how they understood that G1 and G2 are present in addition to this S phase in the interface of the cell cycle. So in this experiment, actually they took these cell lines and exposed to tritiated thymidine. So from this graph we can understand that also. See, so when they exposed for 5 minutes and 10 minutes and 15 minutes after tritiated thymidine addition, this is 5 minutes, not exposure only 30 minutes, sorry. So this is a cells collected of different time periods after exposure. So some cells they collected only after exposure to 5 minutes, and some cells they took that means some cells are taken from the plate after 10 minutes, some for 15 minutes and 20 minutes, 25 minutes, 30 minutes. It's not like from the same plate only. Maybe they might have used different plates, and one plate they observed after 5 minutes, and another plate they observed after 10 minutes, like that. So when they observed at 5 after exposure to 5 minutes, see, only few cells only received the this tritiated thymidine. That means the cells which are at this particular synthesis state received the tritiated thymidine, and very few cells are present in this particular state. So when they expose for 10 minutes, cells which are already synthesized in the S phase, they enter into the next level, and cells which are initially in the particularly in the which are yet to enter, they reach that they are enter into this S phase. Because for 10 minutes, when we give 10 minutes, maybe the cells, when we start the experiment at first in the first minute, so they may be in the condition that in the condition of entering into the this particular phase. Maybe all these cells they enter into here. Okay, and the cells which are already there in the which are in the synthesis, they enter into the even next level, like that. When we take the 15 minutes, even cells which are enter which are to enter into the G1 phase. So I should not mention here G1 phase, because scientists they observed that G1 phase is present after identifying the S phase. So the cells which are yet to enter also may reach to the this particular S phase or synthesis phase when we take more time. So like this, when they increase time, they found like this. Okay, after 20 minutes, see, after 20 minutes what happened. So the cells which are which are divided, that means after a mitotic phase, so what happens, the number of cells increases. So the cells now they enter into the again to this S phase. So when they enter into again S phase, so when you observe the tritiated thymidine, so you find that very few cells again have the tritiated thymidine, because they have to synthesize their DNA. So like this, again after sometimes when they increase the time for again some after 35 hours or 40 hours, they find again this kind of peak. So this is how they conducted the experiment. Thank you.